So I'm very pleased to be introducing Erich Muller for his inaugural lecture. Erich, as most of you know, grew up in Venezuela, which is where he did his bachelor's and MSc in the Universidad Simón Bolívar in Caracas. He then moved to Cornell for his PhD. He went there in 1991 to the group of Keith Gubbins and colleagues and finished his PhD in 1995. He moved back then to Venezuela, where he went back to his former department and moved all the way through the ranks. In fact, sort of almost had a full first career, it seems, in some ways. So he became tenured, he became director of studies, and he was even head of department. He left Venezuela in 2004, or early 2005 rather. He joined us here to be reader in thermodynamics, and he is of course well known to many of the first year students for being the first person that introduces them to thermodynamics. He's actually published a book in this area. It's 25 years old, but still very well known. More about that later on. He's a visiting professor at North Carolina State University, in Cornell, both of Seville's universities, and in DTU in Denmark. He's a fellow of the Royal Society of Chemistry and a senior member of the AICHE. And for those of you who go to the senior common room, you will be very familiar with the journal that he edits called Absorption Science and Technology. Very recently, in fact, I think it's the last round of the Imperial Research Excellence Awards, he was a recipient of that. So I don't really want to be taking away from his time. We've got a busy, action-packed program today, but I know that it's going to be a very exciting and broad-ranging and no doubt stimulating evening. So I'm going to hand over to Erich, who's going to tell us all about jiggling molecules. Over to you.
Well, thank you, Nile, and thank you for coming here. I'd like to start the lecture by actually noting somebody else. I want to talk about Richard Feynman. Richard Feynman was a Nobel Prize winner in physics. And in his book, which is actually, curiously, exactly 50 years old, he made the following comment. He posed himself a question, and he said, well, if in some cataclysm all of scientific knowledge were to be destroyed, and only one sentence passed to the next generation of creatures, what statement would contain the most information in the fewest words? And you can actually think of that and probably have different opinions of what that statement that conveys all of scientific knowledge could be. But his answer was very simple. He said he believed it was the atomic hypothesis. That is, the fact that all things are made of atoms, little particles that move around in perpetual motion, attracting each other when they're a little distance apart, but repelling upon being squeezed into one another. He said in that one sentence, you will see there's an enormous amount of information about the world if just a little imagination and thinking are applied. And this is basically the inspiration for my talk today. What I'm going to talk to you about is about, the title says Jiggling Molecules. It's basically the understanding of the molecular dynamics, or basically what is computer simulation in a nutshell. How can we understand molecules and see the behavior of molecules in a computer? It seems in principle that it might be a plausible statement. It might be plausible, something easy to do. I mean, how difficult can it be to trace the movement of molecules? You can in principle think that we can assign the different molecules a position in space. And for each one of those molecules, we have X and Y, or some number that characterizes their positions. And for each one of these molecules, we can assign a certain velocity. And we could probably, at least in principle, do that for all of the atoms in the system. Now, the atoms don't move chaotically. They apparently are moving around, but they are actually exerting influence one with the other. Atoms feel the presence of the other ones. And just as Feynman was saying, when the molecules are very far apart, they don't feel their attractions. When they come close to each other, they start feeling some attraction. And at some point where they're very close together, they feel a repulsion. And we can express that in mathematical terms by something that we called the interatomic potential, or basically the energy between two molecules. And what you have here plotted is the energy as a function of the distance for two molecules that are becoming closer and closer to each other. If the molecules are far apart, and this scale talks about the distance in terms of reduced units, where each reduced unit is the size of an atom, so this is about 2 atoms in size or in length, you see that there's a very weak attractive force. And the reason for this attractive force is basically that the electron clouds are not fixed, but are actually fluctuating. And this fluctuation in the electronic clouds is what drives this attractive force. As the molecules get closer and closer, the attraction becomes larger and larger. But there comes a point in which the molecules actually feel each other, and these electron clouds start seeing each other and actually overlapping. And at that point, the system starts becoming repulsive in nature. You can't put matter on top of matter, and this is a simple consequence of this fact. Just to point out that if we want to talk about forces, the forces are nothing but the slope of this curve. So you have a region in which you have a negative slope corresponding to a repulsion, and a positive slope in this region, which corresponds to an attraction. So once we have that, once we have all the positions, and once we have the forces, then we can invoke a very old concept corresponding to Newtonian mechanics, and basically the second law, Newton's second law of motion. And this in principle is something well known and well established. And what this Newton's equation says, and it's probably familiar to many of you, is that if we know all the forces that act on a certain system, and we sum them up, and the result of this is equal to the mass of the system times its acceleration. So we could write in principle one of these equations for each and every molecule in the system. I'd like to point out that at this moment, that if we go back to our original slide, you can see that molecules, although you could think of them as interacting in pairs, each molecule is feeling the interaction of all the other molecules in the system. So this sum is a very large sum and has one component for each one of the elements of in your system or in your ensemble. And in fact, you have one of these equations for each molecule in the system. So at the end of the day, what you have is a system of n second-order differential equations in time, which have to be solved simultaneously, and unfortunately, they're all coupled. The math of this is complicated, especially if we're talking about large number of systems. And basically, we wouldn't be able to solve this system of equations in a simple way analytically. So we have to resort to doing it on a computer. And a way of doing that is, instead of solving it analytically, is to take small steps in time. We do this by doing basically a finite difference scheme. And the idea of the finite difference scheme is to choose a small time step. We have, as you saw in the first slide, all the molecules in all their positions, and we think we know all their velocities. And what we do is we calculate all the forces that are in the system. And these forces, as you see, are just dependent on the distances of all the particles. We have all the forces, we have all the accelerations. We can simply take a small time step and guess, or actually calculate, what their position is going to be in a short time step. And we can do that in a reasonably easy way by once we know the acceleration of the molecule, we can know what their next position will be at a given time t. Once they're all in their new positions, of course, the forces are going to be different, because, again, the distances to all the molecules are going to be different. We advance the time, and we repeat this as many times as we want. This, of course, is very boring, but it's done in a computer, and the computers don't get bored, and they just do these things many, many, many times. So we can do this millions of times. And it's a virtue of a computer is its speed, not its intelligence. So the computer won't complain, and will just do it many, many times. Immediately, those of you who have been paying attention will probably say, well, how many times do we have to do this, and how many atoms do we have to consider? or essentially the same question is, how small is an atom? Or how many are there? And how many do we have to account for? The first question, how small is an atom, is probably today can be explained in several ways. One of them is actually looking at them. We are in a privileged position, which most people in our lifetime have not been, that we can actually see molecules. And this is an atomic force microscopy representation of what a pentacene molecule looks like. So this is actually done with a very small tip that feels the forces of the molecules and can actually trace an actual molecule or a group of molecules. And we can see them, and it has, in this case, there's a small bar here, 5 angstroms or 20 angstroms. Of course, that's very impressive when you recognize that an angstrom is 10 to the minus 10 meters. But again, those numbers don't say too much. So you're still taking my word for it that they might be small, but what is small? A dear person of mine looked at this yesterday and said, ooh, that's a big number. But unfortunately, neither Erica nor anybody in this audience has enough span in their brain to understand what this number means. It's just simply too large. This is the number of atoms of water in this glass, plus minus a few. Now even less. But again, that number looks big, but it doesn't convey the actual size of the magnitude of the problem we're talking about. Someone who did answer this in a very nice way was actually another thermodynamicist about 100 years ago. His name is William Thomson, and he was a British scientist, actually the first person to be named, the first scientist to be named Lord. He was named Lord in 1892, became known as Lord Kelvin. And most of you recognize that name as that person as the person who made the statement that there is a point of zero temperature at minus 273.15 degrees centigrade, and that was the absolute zero of temperature. Well, Lord Kelvin had a very nice way of teaching this idea and explaining what this actually meant. He suggested that if we could take all of the atoms in this glass of water and paint them or color them or mark them in some special way in which they could always be identified. And we took this glass of water and threw it to the sea and hoped that all the water in all of the seas mixes perfectly. And then we take another glass of water from the sea. You would have 10,000 of those original molecules in that glass of water. So be careful next time you drink some water, okay? You gotta think what you're doing, what has been there. Okay, so I hope that conveys the idea that the number of molecules that we're talking about is simply impossible to manage, even with any super-duper computer. So what do we do? Well, we're forced with the dilemma of having to look at small numbers of particles. And what we do is we take a small system, for example, like this one is a small cartoon, and we replicate the system in all directions, essentially producing a system of infinite size, which has some periodicity to it. But as long as the system is large enough, we're not really too worried about that periodicity. And some of you can probably see through this cartoon and understand where we computer scientists get some of our ideas. This is actually nothing more than a video game, right? You remember little Pac-Man when he got into trouble Eventually, he would get into trouble with these monsters. He would go out this side and eventually come out on the other side, right? Exactly the same thing that we're talking about here. We're talking about, sorry, I put the wrong, I'm going the wrong way. We're talking about periodic boundary conditions. So we leave out one side and we come back through the other side. Okay, so this basically tells us that we can get away with studying smaller systems. We don't have to talk about these enormous number of particles. We can just talk about smaller systems to give a number, let's say 1,000, 2,000, 10,000, 100,000 particles. And we can solve Newton's equations of motion numerically, and we can do that safely on a computer. So this whole idea is the idea behind molecular dynamics, and it's not a new idea. Probably the first papers that that talk about this actually, again, date magically to 51 years ago. So this is one of the interesting papers that discussed this. You have here in the picture Bernie Alder, which is the one that's standing, and Thomas Wainwright, which is the one sitting down. To my students, I would probably tell them that please recognize that the abstract of this paper has only 3 lines, and the paper itself has only two pages, and it is actually one of the most cited papers in our area. Okay, so there's no need to talk too much or say too much if you've got the right idea. Now, I'd like to point, what they did was actually a molecular dynamics simulation in 2 dimensions of what was 870 hard disk particles. And they did it in the largest supercomputer available at that time. It was actually one of the computers in one of the American research labs. And I'd like to point out to you what Wainwright has in his hand, which is just a copy of many of the sheets he has here. If we zoom into that, he has a graph which shows the movement of the particles as spit out by his computer program. And he recognizes that there's 2 scenarios. There's one scenario in which the molecules move around, basically what we would now consider or call a liquid. There is no positional order. And another region of that box in which the molecules are reasonably fixed in space. There's a nice mesh, there's a nice order to that, and this corresponds to a solid phase. So actually the whole point of the paper was that he was actually being able to see a transition between a liquid and a solid in a computer simulation. Well, that was 50 years ago. And hopefully nowadays we can do better. And the question is how much better? Or how much more can we do? This graph here is what people call Moore's Law. Moore's Law was proposed by one of the co-founders of Intel. And he proposed that every 2 years, the number of transistors in their hardware would double. And that amazing prediction, again made almost 50 years ago, has been held reasonably constant until today. Actually, I plot here the moment I discovered my first computer in the middle of 1980s, and it was actually a small Macintosh, and we were all very amazed by the things that we could do. And this is what we can do nowadays. We look at the exponential increase in computer speed. Actually, to prove that to you, I'm going to do exactly the experiment that Bernie Alder did, but on a phone. Okay, so here's where things can go wrong. So please bear in mind that all the AT works and all the science works, and hey, Steve Jobs, there we go. So my phone, nothing special about it. I run a program which you recognize, it says MolSim. And there we have a simulation, a 2D simulation of hard disks actually done live on the phone. If you take the temperature down, and my hands are a little bit sweaty, you immediately see the formation of a solid, the crystallization, the long-range ordering, et cetera, et cetera. All that we did with a supercomputer 50 years, with the most powerful supercomputer of that time, we can now do with a measly, not even the best of the iPhones. Okay? That's hint, hint. Okay. So, of course, now with our computing power, with a reasonable laptop or a workstation, we can actually look at things like the liquid-vapor equilibrium. We can do a representation of this. We can understand the behavior of interfaces. We can find out what happens we can understand what happens on a macroscopic scale by looking at the microscopic scale and understand and recognizing that these macroscopic phenomena are nothing but a reflection of what is happening with the molecules themselves. Again, another question that you might ask is, well, okay, now if you could do that with an iPhone or as a small machine, what can you do with those big, powerful supercomputers? So here is a world record. Actually, it was a world record until not recently. It was a simulation of a cube of copper, or Leonard-Jones atoms, actually, in the order of 1 trillion atoms, or 1 American trillion, so 10 to the 12 atoms. And it was done for a very short time span. However, it's interesting to note that the macroscopic size of this box would correspond to something actually, something that you could see. It would be around a micron in the order of the micrometers. So it would actually, you could see it with for example, a common microscope if you're talking these sizes. I'd like to make the point that this is not the way to go. This was done on with over 200,000 processors in the most powerful computer at that moment. It is a good world record because it lasted almost 5 years. It was broken by one of our ex-postdocs, Martin Hirsch, this year. And you can see that he didn't advance too much, right? He advanced by a factor of 4, but of course now he's a world record holder, and that's good enough. So the rest of the talk, I'm going to now talk a little bit about now what can we do with this power, what can we do with these tools, and what are the types of things that we can explore, and where can we go. And I'll start with something reasonably familiar with soaps and bubbles and water and taking a bath. In general, when we have this type of situation, we have water and usually an oil phase, and we add a third component, which in this case is a soap, is an amphiphil. And when we add this amphiphil usually stays at the surface between the air and the water. If we keep adding surfactants or soap molecules, and these little colored guys are the soap molecules, you see that eventually these molecules start self-assembling in very unique shapes called micelles, very spherical molecules which start separating from the interface and entering into the bulk liquid phase, which in this case is pure water. So let's take a closer look at one of these micelles and see what we're talking about. Most surfactant molecules are made of 2 parts. One part, which is usually called the head, is a lipophilic or oil-loving part of the molecule. And it's joined, or physically joined, to another part, which in this case is called a tail, or a hydrophilic tail, which likes water. As you know, oil and water do not mix. So this particular molecule has one part of 2 behaviors, and actually the molecule itself doesn't like itself. So this part hates the other part, but they're joined in marriage and they can't divorce. So that's it. That's your life. Particularly, this one is called Triton X100, which is just a fancy name to just show this type of surfactant. It's a very common surfactant found in most very strong detergents or cleaning agents. And what happens is that upon putting this in a hexane or an oil-water mixture, basically water gets sequestered into a micelle, and the surfactant, or amphiphilic molecules, sit themselves in the interface, separating the water from the hexane. So this is a fairly a standard or well-known situation. Now I think it's time for a small break and actually see these things in action. Just a small health and safety moment. I do not wish to be recorded without doing the proper. Yeah, I've got this or two. The problem is I can't see anything. So I brought a small demo to show you. In this flask, I have on the top part I have hexane, which is transparent, and on the bottom I have water, which I just tinted with a little tint of blue just to make it look nicer, and so you could have a little bit more of contrast in the back. So as you know, I mean, no matter how much I shake this, and no matter how long a time I spend doing this, these things don't mix. You know, this is a classical reason why, for example, people like my lovely wife uses a blender to make mayonnaise, right? She takes water and olive oil and puts, now in this case, egg, which is the surfactant, blends it all together, and then you get an emulsion, right? So that's exactly what we're going to do, and just replicate that. So I'm just going to add a surfactant. Let me try to get the correct one. And so there's nothing special about this. This is the Triton X100, so it's just a fancy name. So if we add this one, and again, give it a little shaky. There we have the equivalent of mayonnaise, an emulsion. Okay? What happened was now it's turbid. Right? It's not clear, it's not transparent. Those little micelles over there, the ones we had before, are about the size of the wavelength of light. So they scatter light, and then of course it becomes non-transparent anymore, and now we see it as an emulsion. But rapidly you recognize that the emulsion is not stable. Okay? If we wait long enough, these phases will start separating, and eventually we'll go back more or less to something that we had before. So it looks nice, but in terms of food or mayonnaise, it's definitely not satisfactory. So what can we do? Can we do something better? And this is the whole point. The challenge here is to be able to reverse engineer molecules in such a way that we make them do what we want. Can we build molecules that perform in a satisfactory way or perform or have the behavior that we want? And this is the whole point or the whole ethos of some of our research, is to try to model in a computer or try to play with our computer models in a reasonably intelligent way and try to find molecules that can do what we want and that can behave as we want and can reproduce the behavior that we want. So one of these special molecules is now this one. Again, it has another fancy name, AOT, and for those who really love the chemistry, it's a big fancy name at the bottom. So let's try the same thing with AOT. Again, no tricks here. This is exactly the same setup, no magic. And its phase separates exactly in the same way because it's water and hexane. So now let's put AOT on this. Top of the wrong one. You can give it a little shaky to get everything in. And this takes a little bit of time. But the first things that you're starting to see is that now it's much more transparent. And if we leave it for a while and get everything mixed up, you'll actually see that the two phases have disappeared, and this thing is almost completely transparent. This is what we call a macromolecule. We'll leave it a little while there, and actually those who are bored can actually keep looking at it, and actually will evolve into a very transparent phase. What's happened now? Well, this is a microemulsion. Now the sizes of these micelles are much, much, much smaller, even much smaller than the wavelengths of light. So actually light goes right through it and looks like a transparent, beautiful mixture. The color, of course, is just indicating that there is a little bit of water, of tint, and the water is there. But now you can sort of convince yourself that it's just a one perfectly transparent one phase from those two phases that we started. Okay? So that is a very simple example of molecules self-assembling, or basically molecules forming different phases and different structures on their own without basically no external force. I mean, we're not saying anything to them. They're not intelligent beings, right? They just do what they have to do, and they assemble in their own way. But this idea of self-assembling in molecules is extremely important. It's actually the key to everything. DNA, for example, is a classical example of this. The DNA molecule actually breaks down and self-assembles on its own. And what you have here is a small video that talks about an enzyme, or shows an enzyme, a helicase enzyme. And this enzyme is breaking a DNA strand. On one side, it's replicating the DNA strand, and on the other side, it has even a harder time because the DNA strand is backwards. So it has to actually invert the DNA strand and fill it in. So from one DNA strand, this complex enzyme is obtaining two different replicas of that original DNA molecule. Now, you have to recognize that there is nothing that we could call living here. These are just chemical and physical interactions. These are just molecules which are just assembling and reassembling due to the conditions in which they are around. And that is actually the basis of all of life. So if we can harness this potential, if we can look into this potential and try to work out how molecules work and how we can tailor molecules to our own needs, then we're probably going to be able to do some pretty interesting things. I'll start with a simpler model than that of DNA. This part is about Bola amphiphiles. It's a very big name. An amphiphil we just saw is a molecule that has it likes everything. Okay, amphiphilic likes everything. The word Bola amphiphil implies that the molecule itself is a very interesting one, and here is a replica, a molecular model of that. The core of that molecule is a liquid crystal. It's a very, very straight, very rigid molecule, like a pencil-like, which in the extremes has polar groups. So these groups are very hydrophilic. This part in the middle is actually very lipophilic, so very oil-like. So if we chop this down, this would be an amphiphil, but this one is a Bola amphiphil because it has polar groups on both sides. But we make things more complicated by decorating this molecule and putting a chain. And this chain that looks like grapes on it is actually representing a fluorinated part. Now, the point of this is that in this molecule, now we have three different behaviors. We have a liquid crystal, or a part that loves the oil, a part that loves water, and a third part which hates these two, and so it's in the middle there. So now it's getting more complicated. Now we have a system which not only a molecule that hates itself three ways, and it poses quite an interesting challenge, because this type of molecule produces very rich phase behavior. This slide shows some experimental results from the group of Carsten Scherke in Germany. And they've used this molecule and analyzed it. They've synthesized this molecule. Actually, it's a synthetic molecule. It's not it's not something found in nature. And they did something very interesting. They took the original molecule and started synthesizing it with different chain lengths. And for each one of these chain lengths, they found out what their behavior was. For the molecule without the perfluorinated chain, what they could see were smectic liquid crystal phases. That is more or less the same thing that would happen if you throw pencils in a box. They'll start aligning themselves. So that's the type of behavior they would see. If the chain was too long, then they would start seeing the behavior classical of long polymers. Lamellar phases, very similar to the ones found in biological scenarios. However, for intermediate chain lengths, they start finding some very strange behavior that would correspond to 2D arrangement in terms of tubes of different lengths and different sizes. So they've characterized that experimentally and basically through the problem at us. So we took this molecule and mimicked it in a simpler fashion, maybe not as a detailed description as we would want here, but recognize that the molecule has some groups which are important, the polar groups at the end, some rigid structure in the middle, and an alkane chain, a perfluorinated chain at the side. Now, you might wonder why I did this. I mean, if this was very nice and very descriptive and all the atoms were in its place, there is a reason for that. And the reason is that we want, basically in this type of research, we want the results for today and maybe not for next year. You see, if we take a simulation of these types of molecules, we have over 100 atoms for each molecule. And we need to have a reasonable system size, around 1,000 molecules, to have the correct behavior. And if you start multiplying the numbers, you recognize that rapidly you need at least 10,000 million calculations for each time step that you want to do. And usually in some of these simulations, you want to perform millions and millions of time steps. So again, you add another million to this, the number again becomes one of these enormous numbers. And no way to do that in reasonable time. So what we do is we lump together some of the groupings into what we could call coarse-grained groupings, some larger superatoms, a much smaller number of atoms, much simpler potentials. And by doing that, we simplify our calculations by 6 orders of magnitude. And that is serious speed up. In terms of waiting for that, we would have to wait another 50 years to have the computers to do that. And we can do that with our intelligence rather than with our computers. In terms of the potentials that we use, or the intermolecular potentials that we use, in general, there's no need to be super specific about this. And the general behavior of amphiphilic molecules is governed by the fact that they hate or they love each other, not by the absolute specific details of each one of these interactions. We have in the group done a lot of effort to map these intermolecular potentials to macroscopic properties, and we know how to do that properly. In this particular case, it's an oversimplification of things. And the red spheres are very, very much like each other and hate everybody else. The blue spheres, which are the perfluorinated chains, like themselves a little bit and hate everybody else. And the white ones, I guess, are the non-social ones that don't like anybody. But so it's a very simple scheme, potential scheme, which can, I repeat, can be mapped into realistic behavior. And we can now do our simulation in real time and see what happens. So we can take one of these systems and put them in a computer, in a box with our periodic boundary conditions, start with a very well-mixed system, and lower the temperature. And once we lower the temperature, we can see what happens. As time goes on, we can see here what an order parameter, which describes visually what you're starting to see, that the molecules are starting to self-assemble in a unique way. And when we reach the 20 nanosecond point, there will be a jump in the structure, and it will become very nicely, beautifully ordered two-dimensional tubes forming a very interesting phase. This is not a solid. This is a liquid crystal. This is a fluid. If we move it, it shifts. Okay, it's actually a liquid which has structure, very much like the screen in your laptops, which you put a finger in it and it kind of moves around, or some of the phones we have. And we can do, of course, this with different chain lengths and different scenarios and different temperatures and map out the whole phase diagram. And not surprisingly, we were able to map exactly all of the phase behavior that was seen experimentally, even predict some of them as the triangular phases, which had not been seen in the experiments and were later seen after we published these papers. They were actually seen in the experiments. What is probably more interesting or sparks a mind is that we can use these for novel applications, for things that people haven't thought about. And this is a preview of something that you haven't seen, that is not anywhere around. And it's basically a self-healing liquid crystal membrane. So this is a membrane which allows the passage of a certain species, but instead of being a physical membrane like a paper or like a plastic, it's actually a liquid crystal. It's movable and it's tunable, and you can actually change its depth by simply changing the mass or removing mass from it, or you can simply make it disappear by adding another solvent or by heating it up, and it will all disappear and become isotropic. And this starts opening up a whole new area of novel material applications, and again, the possibility of reverse engineering molecules to try to do what we want them to do. I'll give you another example before I finish. This is a paper from a group in the U.S. Which actually again designed and built these molecules without really knowing what they would be good for. I mean, there's an effort in synthesis here of producing these types of molecules. Upon looking at the micrograph of these molecules, they produce these very, very beautiful spherolytic structures, meaning that there is an order and there is a liquid crystal ordering in these molecules. This molecule is made up of a polar head group, a rigid intermediate group, which is very benzene-like or phenyl-like, and it has long tails of hydrocarbons. So it has roughly the same elements of our first Bola amphiphil, but the shape is different. Does shape matter? Well, the experimentalists seem to say it does. What comes out of the literature says that if the molecules are very flat and look like a pizza wedge, well, they'll form little stacks of coins or columnar phases. While if the molecules are a little bit larger, they will look like little cones, and these cones will form spherical phases. And this is what comes out of the literature, what comes out of reading the experiments. And we wanted to challenge that idea. We wanted to see if the molecular modeling could teach us a little bit more about these molecules and how they behave. So again, following the same logic and the same idea, we developed a coarse-grained model which has exactly the same behavior. It has a head group which is very hydrophilic, a core which is lipophilic or basically rigid, and a chain, a tail, which would represent the alkyl chains, or again of a different nature. So again, yellows and greens and reds have different interactions, and they hate each other. Again, they're all in the same molecule. And again, we can do our simulations and see what these phase behaviors are and what they're all about. So to start with, we ran our simulations at high temperatures. There was a lot of energy. Everything is mixed and everything is easy. And there is just a single phase. There is no structure whatsoever. All the molecules are just moving around. We see absolutely nothing. If, however, we lower the temperature, we quench the system, immediately we start seeing one of the first types of behaviors that was suggested. However, if you remember, this small molecule was supposed to form columns. But no, they form spheres. And these little spheres move around like they were a liquid, but actually they're not fixed spheres. They're liquid crystals, and molecules will move from one of these nucleus or spheres to the other. And most curiously, they're all of the same size, which is quite unique in nature. This is something that's not seen in nature commonly. If we raise the pressure a little bit, this liquid crystal phase, or this phase of spheres, actually orders itself like if it was a crystal. And it looks like a solid. It has a very large, very nice structure to it, but it's still a liquid crystal, and it budges. So if you move it, you deform it. And if you if you heat it up, again, it becomes a liquid. If we keep pushing the pressure up in this system, what happens is that then we start obtaining the phase that we were expected to see, the columnar phase. The spheres join up into layers or worms, and you can actually see a columnar phase forming for this for the same molecule. So while we did prove that shape does matter, and it makes a difference, we also proved that the thermodynamic conditions at which this system would be play the most important role. And understanding the relationship between order and energy, and what is called entropy and energy, is actually the key to studying these types of systems. So we're coming close to the end. And well, what I just wanted to say as an epilogue of this is that there are profound questions in nature for which we have no answer whatsoever. What you have seen in this lecture is just but the tip of an iceberg, and an indication that even the most complex aspects of nature, of physics and biology, all boil down to the description of matter in terms of very simple interactions, in terms of this jiggling of molecules. And we were very happy in our group and in our community by news that happened last week, in which the Nobel Prizes for chemistry were announced. And the winners of the Nobel Prize in chemistry, which probably for some of you don't mean anything, were actually computer chemists, and they worked in the same area that we're working, and they were the ones that developed some of the potential models that we use in every day. One thing, of course, I'd just like to point out in this slide, which is very interesting, is how Warshel is using his two laptops, one on top of the other. And so this, I know many people who use several laptops at the same time, so I guess you can now relate to some very important people. Of course, I could not finish without talking actually about the people behind the molecules. Of course, all of what I've done and all of where I've achieved could not be possible without the help of a very large army of people which have in different points of my years as an academic helped around. Some of my research students, which have been helping out in different aspects of everything, that have helped write the papers. Also, some of my collaborators, some within the countries I've been, or actually in many parts of the world, in Germany, in the US, in parts of Latin America. And of course, of my colleagues, and I of course apologize for some of the ones that are not on the list, but the list goes on, and believe me, I haven't forgotten you, even if you're not on the list. A special thanks is to my MSc students, which have spent longer than usual time with me, and have tolerated me, and actually even more for my PhD students. And here is a picture of Aladin, actually my first PhD student. Actually, he's I don't know who's happier. And of course, all of my current and future PhD students, future PhDs, which I'm sure that they'll all get their degrees. Also, of course, the postdoctoral associates and visitors that we've had along these years. But of course, nothing would be done without mentoring, and I give us a very warm thank you to some of my mentors, Keith Gubbins and Antonio Esteves, which curiously are in this picture together. And this is me when I was younger, actually. Actually, even a more special thank you to my dear colleagues and friends of the MSE group. And of course, last but not least, my loved ones. And thank you all for your attention.
on the list of Eric's mentors, you saw Norman Carnahan. I would like to hand over to Norman now to give the vote of thanks.
Thank you, and especially thank you, Erich, for such a wonderful presentation on such a diverse area of application, or realm of application of how molecules jiggle. And my remarks today, when I was trying to assemble these, I was thinking back on some of the very first times that Erich and I had interaction, actually prior to his going for his doctorate in Cornell. He was still a young teacher in the Universidad Simón Bolívar in Caracas, and was already stimulating his students or infecting them with what I lovingly refer to as van der Waals itis. And van der Waals, like our common mentor in literature, Richard Feynman, was always looking at things in terms of very fundamental interaction of repulsion, attraction, and as Erich and I later worked together, we also throw in the geometry of the molecule, because there are certain steric phenomena that happen that are not strictly due to the basic forces of repulsion and attraction. I also want to recognize that we have some visitors from far away, Professor Masters and Sifferstein from Manchester, and Professor Niemark from Rutgers University in flooded New Jersey. But again, I look back over the years that Erich and I have known each other and have worked together in various ways. What you see today, his enthusiasm, this infectious approach to the science of thermodynamics, is exactly what I saw when I first met him in 1988. He became infected, or he was already infected, I'm not sure, with the van der Waals itis, because we had a very, very long talk at our very first meeting about equations of state, about molecular phenomena. My encouragement to him and to all young students and colleagues is to always look at things at that very first level. All of the things that we see, and as you're seeing now with this visualization, result from these fundamental forces. And many of my engineering colleagues work in a realm of macroscopic application. They miss such beauty that you see at the fundamental level. And all of the problems that I have seen in industry or in the applications of science really begin and are best treated if you can define the fundamental physics of what is going on. In the time of the 1980s, the computer that Erich referred to, or the Commodore PET that I started with, couldn't do these things. We were just at the beginning. Professor Gubbins was one of the pioneers in trying to convince people that one day we can actually do this. And there was a lot of doubt in the early days of the, let's say, 1960s and 70s as to whether the computers would ever be capable of doing these things that we saw today. But Erich has been a major force, I think, in advancing this. When he went to Cornell and studied with Professor Gubbins, he came back to Venezuela and rapidly went from professor to head of the department at the Universidad Simón Bolívar. There were a lot of things that happened since then, a lot of unfortunate things that happened to a beautiful country. But the fortunate thing is that he was able to come here to this great university and continue his career. On behalf of all of us here, Erich, I want to thank you, and I can forecast a long and productive career at this campus, as I've been told, because some of them, George had expressed a concern that perhaps now that you've gotten to this level, you'll be going free agent, but I encourage you to stay here. All right, well, thank you very much.
Okay, thank you very much, Norman. So, Eric, I might need your help here. Can you bring up? Excellent. Yeah, you might notice we've got an unusual AV setup today, so our whole console failed today. So I certainly want to thank the AV people from Events who managed to rig the system up. We're actually hardwired from Eric's computer through to the projector, so apologies for the people who have to sit over there. So Andrew made me promise to not let Eric get away completely lightly and to tell you some other things about him that you may or may not know, some of which I'll be talking about for a couple of minutes is fact, and I would say some is a bit of speculation. So here, this is Eric, this is him in Cornell, so you can just about see that he hasn't really changed very much, but that was way back in those days. So here he is again with his supervisor. I'm not sure where that's taken, but again, you can see a very relaxed young man. He's just taken over as Director of Resources, so I think that may or may not change in the next 5 years. This is Eric when he's just, I think, found out that he's passed. Is that right? Yeah. So he didn't look too stressed, did he? It doesn't look as though it was an ordeal. And this is the evidence of his PhD. Notice the non-English spelling, which is something I'm going to come back to in a minute. And here he is again, sort of celebrating his graduation as a PhD. So that's way back in 95, I guess that is. So as we heard, he moved back home. And you can see, what a lovely campus it looks like, and what a sort of, again, he looks like a very sort of relaxed and refreshed young man there. So during this time, he wrote a book, and for those of you, who have taught first years, you know that sometimes when you introduce them to books, you get an interesting response. I don't know whether this is a positive or negative response, but this seems to be how the book was received back in Venezuela. I think because it's champagne, it's considered a celebration, but it's not a celebration style that I'm familiar with here, but maybe it's one that we should be introducing. So, yeah. Oh, here we see someone else who appears in the frame. So this is probably some of the early collaborations. So most of you will recognize a young George Jackson there. Again, I don't think time has taken its toll too badly, actually. So of course, from what we've heard today, Eric is a great conceptual thinker, and of course we know him very much as a theoretician and modeler. But what you don't know is he's also does a lot of empirical work. So this, for example, is his attempt to brew alcohol at home or in the lab, I'm not sure which, and probably interested in measuring some properties there to see whether or not it has met the design requirements. And here he's interested in this phenomenon of solid-liquid phase transitions. But it actually ties very well with his general interest that you probably have heard about, which is in food and drink. But an area that you probably don't know about, and which again is quite interesting, is the fact that he thinks much more widely, which I think he hinted at in his closing words of his lecture, that there are many other systems which, like molecules, can be described by these different kinds of forces. And some of the systems he's interested in is human social systems, and how emergent properties in these kinds of systems can actually be described to some extent by these phenomena. So I was really interested, and I started to read about it. And then something else caught my eye, which is this bit here, which is something completely different, which is that he's a Venezuelan-born English chemical engineer. So I scratched my head and I thought, this must be just sloppy language. It can't possibly be the case, because we all know him to be sort of very exotic Latin American guy, not really English. So I thought, it's probably almost certainly just written badly, and what they mean is obviously he's working in England now. But I thought I'd just do a bit more digging around, just to see if I could find any more evidence that perhaps he's been conning us all along. So the funny thing is, I did actually find some evidence. So first of all, I've actually found out he's a world expert in crisps. Now, how English can you be? So he's actually got this article here, which you can look up in The Guardian. So the Observer food critic, Erich Muller, and of course, and Shane Osborne, sort of comparing different kinds of crisps. So I think he's either gone native or perhaps there's something he hasn't told us. He also likes to sort of act the mad scientist sometimes. So this, I think, is the Imperial Festival. We'll see more of this in a minute. And he likes to attend famous sporting institutions. So this is him at the 2010 Wimbledon. So I was just beginning to become a bit more convinced by this hypothesis. And then I found out that he also attends Doctor Who conventions, which really, how English can you get? So I want to leave you with a video, which again, Eric may have to click for me, but I'm going to call him up anyway to thank you very much and thank him very much. So thank you very much for attending. And what we have here is a memento of your excellent talk today. I think it gave everybody a lot of food for thought. We enjoyed it very much. So this is yours to keep.
Oh, great.
And if you want to set this off.